Right, welcome to the first session, the technology session. We have quite exciting talks here. They have to do with the developments in technology, both on the experimental side, but some of the talks are going to focus on the computation, so hopefully it will be interesting and exciting for everybody. Our first speaker is Christoph Bock from the Center for Molecular Medicine in the Austrian Academy of Science. He's going to talk about CRISPR sequencing in single cells. Please, Christoph.
Thank you for inviting me here. Great pleasure to present our work. We are combining experimental technology with computational tools. And I would take the analogy a bit further today. We want to understand cells also as programs in a way, as little computers that have encoded information that are programmed to do certain things. And in particular, we want to take a perspective of the past and future of cells, how cells developmentally become what they are, and through that are pre-programmed to pursue or to react to stimuli in certain ways. So you could argue that's a form of epigenetics in its more original form. So it's not just DNA methylation or histone modifications, but it's really this idea that cells live on a landscape that has some conceptual similarities with a potential landscape where cells maintain the ability to go in certain directions, but are more restricted in other directions, summarized by the Waddington landscape, which really captures a lot more than we commonly think about it. It captures already that transcription regulation and epigenetics are really two sides of the same medal. So if you look from the top of the epigenetic landscape, you see epigenetic states. If you look from the bottom of the epigenetic landscape, like here on the bottom right, you see the regulatory, the transcription factors, the regulatory mechanisms that keep it all in shape. And then the moment you start cutting these ropes, like as it is happening in cancer, the landscape changes and you suddenly get yourself stuck in areas where they should not normally be in. Now, when you're a computer scientist, like I am, if you're looking at these type of drawings, some of them very old, you immediately want to model that. You want to find a quantitative predictive description of these epigenetic landscapes, and ideally convert this into a problem where you can do mathematics on cells. You can predict how cells might be reacting to different perturbations, how cells differentiate, how cells maintain their plasticity. And with single-cell technologies and everything that's related to it, finally the technology is there, and I would say the data are there. Just the concepts and the mathematics are just not quite good enough yet to really infer and make actionable this type of landscape. So this is the theme. I don't say we have a solution for it, if only very partial ones, but this is certainly what motivates us. In the first two parts of the talk, I will take examples from immunology about reconstructing the cellular past and future. And then in the third part will be cancer, and finally a bit of an outlook what perturbations really mean for dissecting high-throughput biology in single cells. So we first got into the topic through kind of a narrow sense, single-cell epigenetics, studying DNA methylation at the single-cell level as part of the International Human Epigenome Consortium, where we found that based on DNA methylation profiles, we could reconstruct computationally using relatively straightforward machine learning methods the developmental lineage tree that underlies human blood development. So in a way that was a confirmatory project because we already knew what to expect, but we validated this technology and then could take this technology forward to diseases where things go wrong in very odd ways. And one disease I got interested in back then through collaborators at the St. Anna Children's Cancer Research Institute in Vienna is Langerhans cell histiocytosis, which forms these lesions in the skull, on the skin, etc., that are essentially accumulation of immune cells. And it's a bit of a hybrid. It's not quite a cancer, but it's not quite an immune disease either. It has a bit of both. It's driven by a strong oncogenic mutation, but it never gets into this process of somatic evolution that characterizes cancers. So we looked into these lesions where the pathologist basically told us this is just the same cell over and over again. And we used these technologies to look at whether there might be kind of a developmental lineage tree hidden. And indeed, if you look at the single cell level and you apply computational methods, in this case, this was based on the concept that entropy is associated with cell differentiation state, you indeed find a kind of a whole developmental lineage, an apparent developmental lineage in these lesions that feeds the disease. Now, you can also take that further into clinical trials for such diseases as loosely related diseases, sarcoidosis, essentially a formation of again, an accumulation of immune cells in the skin in different organs that causes quite some problems to the person that has it. Actually, this is one of the most common side effects of tattoos. So if you have large tattoos, the macrophages sometimes get activated and start forming what you can see here under the rose. There's these patterns. These are essentially macrophages that start trying to encapsulate basically the color into something like in the same way as the body might kind of decide to encapsulate a pathogen that it cannot get rid of. And then this creates this kind of self-promoting inflammatory lesions. And this is a disease for which there really no good causal therapies exist. So a lot of interest was coming up when a colleague in Vienna found that mTOR signaling appears to be important for this type of disease in a mouse model. So together with clinicians, we set up a crossover NF1 clinical trial in which we do single-cell sequencing as well as spatial profiling at several points over the course of the clinical trial. So practically speaking, a patient enrolls in this trial, gets a biopsy of the skin, then goes on either topical treatment, so a form of cream, or systemic treatment, a pill for this type of mTOR inhibition using kind of approved mTOR inhibitors. And then in between these different phases, we always take a biopsy and we do single-cell sequencing. Now, this trial is ongoing, and I cannot yet give results of the trial, how well it has worked, but I can talk about the ground stage profiling. So at baseline, when the patients first enrolled in this trial, we did the single-cell RNA-seq, the spatial profiling, and then also a lot of immunofluorescence imaging, and we could reconstruct what is going on in these lesions. This time not only at the single cell level, like in the Langerhans cell histiocytosis, but also on the spatial level. And essentially what we reconstruct here is not so much disease lineage, but an ecosystem of cells that kind of together maintain the inflammatory environment of these particular lesions. Driven by macrophages as really the pioneers here, T cells that then are then attracted and add to the inflammatory character, but also fibroblasts that provide kind of stromal support for these lesions. Speaking of fibroblasts, we usually think of immunity in terms of the innate immune system, the adaptive immune system, and we simplify it to basically the myeloid and the lymphoid lineage. So T cells, B cells, or the adaptive immune system, macrophages, dendritic cells, etc., the innate immune system. But a postdoc in my lab kind of felt that we were overlooking by essentially talking about immunology mainly from the perspective of hematopoietic cells, we overlook what is actually a well-understood contribution of all the cells of the body that all have some capability of innate immune responses. So he sorted from mice 12 different organs and extracted from those organs the structural cells, the building blocks of our body, the epithelial cells, the endothelial cells, the fibroblasts. And he found a kind of a surprising degree of immune regulation in there. Even more so at the epigenetic level than at this transcriptional level. I think an analysis that is very useful and sometimes underutilized is plotting promoter chromatinase accessibility against gene expression, in particular when you couldn't do this over timeline or in response to a perturbation. And what we saw here is that a lot of these structural cells maintain kind of overly open promoters for immune genes. And so why would cells invest a lot of energy in keeping the promoters open of immune genes that they don't normally use? We argue that this is a form of cells maintaining potential for rapid immune gene activation. So a structural cell does not need to constantly express immune genes, but it needs to be ready to rapidly activate those genes when an infection comes about, and it needs to kind of interact with the hematopoietic immune cells to mount an immune response. So obviously that's a hypothesis that needs testing. We tested this together with my colleague Andreas Bergthaler using a viral infection model. So we challenged these mice with a systemic viral infection and indeed saw what you can see here on the right is kind of something similar to a ROC curve, essentially showing how much those genes that have a pre-activated potential, epigenetic potential for upregulation, indeed upregulate upon challenge. Now you see, if you look at this from a perspective of a ROC curve, that's clearly better than random, but it's also not particularly strong. And that makes total sense because we are challenging these cells in one way here with a viral infection model. But there's so many ways that cells need to be prepared to react upon. Viral infection, bacterial infection, physical insult, wound healing, all of these aspects. So kind of what we are arguing here is the structural cells of the body are not just mere building blocks, but are highly dynamic contributors to immune cell regulation, enabled by an epigenetic potential for rapid activation of those genes that are needed to respond to such stimuli and a diverse range of stimuli. So taking this a bit further to cancer, following a quite similar approach as for the sarcoidosis, essentially following patients over time, and in particular also when they go on treatment, we can really try to map out this idea of cells having a developmental history and then a future potential that very much defines their disease, also defines how they are going to react to therapies. The example here is chronic lymphocytic leukemia, which is a very interesting disease because in some patients it stays indolent for 20 years, so people can live a reasonably good life with that disease, 10, 15, 20 years, and then die of something completely different. But in other people, the disease very rapidly progresses. And we have the additional advantage, and other people have shown that these leukemic cells really keep an epigenetic memory of the stage of B-cell differentiation from which they were originally derived. So we can use look at the epigenome to reconstruct where these cells were originally derived from. But our focus here was kind of looking prospectively. So when these patients go on treatment, essentially a treatment with ibrutinib, a BTK inhibitor that shuts down B-cell receptor signaling. So these leukemia cells, they are addicted to constitutively active B-cell receptor signaling. And when you take that out, things happen. And we followed those things in a very small cohort together with collaborators in Budapest. and looked at the kind of time-resolved epigenome and single-cell transcriptome profile in these patients, applied Gaussian processes for computational modeling, and found a shared program that is executed on these cells. So the moment that you take out the BTK, the B-cell receptor signaling, we see a kind of shutdown of kind of a loss of NF-kappa B binding to the genome, and that seems to be essential for maintaining them in their these cells in their aberrant state. So we see a rapid shutdown of lineage transcription factor binding to the genome and an erosion of leukemic cell identity. And then those cells they are kind of in a no man's land of the epigenetic landscape. They are in a forbidden space, and they should rather go into apoptosis. But for some reasons, they do not. They go into quiescence, which is better than nothing, because that is enough to make that acute disease and aggressive disease in some patients chronic. So as long as you treat the patients, you essentially push those cells into this quiescent state. But the moment you remove the therapy, they rapidly come back because these cells are still there, and they can kind of, the moment you let them recover the B-cell receptor signaling, they also kind of regain their consciousness in a way and are able to reconstitute the leukemia. So we combined this here with a chemical screening setup, imaging-based chemical screening, in order to find drugs by comparing patient samples before and after the onset of therapy, and then do drug screening on those to identify how we can perhaps, with a double hit, bring those cells from the quiescent into an apoptotic state. Now, this is early days, and much more work is clearly needed, but to me, overall, this perspective of diseases over time, progression, developmental history, future potential, etc. The real question this poses is if we are thinking enough of diseases as temporary kind of processes over time rather than just single snapshots. Obviously, a patient comes to the doctor at a single point in time, and often our single cell data is at a single point in time, but wherever possible, we should really go for actual time series. Pseudotime and pseudotime-temporal ordering are great tools that can achieve a lot, but really we want to follow patients over time series, clinical time series, including how they react to a new drug. And then perhaps eventually we can do enough modeling of this landscape to predict how patients react and essentially play a form of evolutionary chess with a tumor, where you take a therapy forward, already calculating ahead how the tumor will eventually overcome this therapy, and rather than using up your options one after the other, try to use combinations, take them offline again when you see first indications of resistance, and really adaptively treat patients without the goal of eradicating the cancer, but with the goal of keeping the cancer sufficiently under control that eventually you gain those years of life that may be enough. This is, especially in the elderly, the goal doesn't have to be to cure the cancer in the sense of eradicating every single cancer cell. The goal can be, in many instances, to allow a certain kind of chronification of the disease living with the cancer. Now, in the last part, this is perhaps the most technological part of the talk, is the question how we can go from this descriptiveness of what I talked about so far to an actual causal mechanistic conclusions. Time series is already a big step, because there's even this concept of Granger causality, that earlier events can cause later events, but later events cannot cause earlier events, that at least show that there is a loose relatedness between time series and causality or causal modeling. But eventually, perturbation is what helps us to get to causality. And I would argue that perturbation needs to be scaled up from one at a time, one gene, one postdoc, to at least dozens or hundreds at a time in a given system. Otherwise, we will always go for the kind of the safe bets and do more work on P53 and more work on the usual suspects. I'm all in favor of the usual suspects. They are important, but I also think that there is a huge kind of landscape that we have just not investigated because it would be too risky to send a PhD student or a postdoc down that route with very little preliminary data or very little knowledge or tools. Fortunately, the methods for perturbation analysis are becoming ever more powerful and useful. And it's really the combination of CRISPR screens, computational methods, in particular deep learning, and organoid technology that are coming together to form a way to do perturbations at scale. Now, I'm just presenting building blocks here. I'm not saying we have it all sorted out, but I think these building blocks, based on these and related building blocks, we will see a lot more kind of high throughput perturbation, high throughput causality work over the next couple of years. Key point is kind of the evolution of CRISPR screening. CRISPR screening originally was really about finding the one hit and then working that up. If you look at a lot of very good CRISPR screening papers, the screen is really figure 1, and then figure 2, 3, 4, 5, 6 is working up the mechanism of that screen. That's great and a very powerful approach, but the more we can pump information out of the screen itself, the kind of more we already know the longer we can stay systems biological before really going small scale. And the power here lies in combining CRISPR screening with single cell sequencing, with FACS sorting, eventually with mass spectrometry perhaps, certainly with CyTOF, as a way to read out what's happening in single cells. So we wrote a review article, essentially a primer, like an introductory review that's linked here on what we call high content CRISPR screening, with the idea that the screen should not just provide a list of targets, but should already, together with each of the targets, give an indication of potential mechanisms. And clearly, our CROP-seq methods or the perturb-seq methods, CRISPR-seq, Mosaic-seq, and several other methods that have been developed to do CRISPR sequencing with single-cell sequencing readout have become a very powerful tool here and are widely used, even for genome-wide screens now, but for the most part for kind of candidate screens in the range of a couple of hundred to a couple of thousand target genes, and as validation screens. So when you do a screen, rather than picking based on biological knowledge from a long list of potential hits, kind of a validation screen with single cell technology can be a very useful method. Now, what this technology has suffered from, and why CROP-seq and perturb-seq hasn't really replaced CRISPR screens as we know them, is clearly because of the cost of single-cell sequencing. So if single-cell sequencing were free, I would think in pretty much any screen you wouldn't really do a classical kind of just survival screens anymore, but you would always want to know as much as possible about the perturbed cells. Unfortunately, single-cell sequencing is very expensive, and CROP-seq screens are very costly, too costly for some applications. So we invested quite a bit of time to make single-cell sequencing cheaper, essentially by combining droplet-based approaches with combinatorial indexing in such a way that you first do one round of combinatorial indexing, and then you do one round of droplet-based encapsulation on the 10x Genomics system or any other single-cell platform, and you get much more high throughput readout, essentially scaling from kind of 5,000 to 10,000 cells in one 10x lane to, I show data here, 450,000 cells. So that's a very powerful approach. There's two downsides to it. The first one is you have to fix your nuclei, and that does reduce the quality a bit. And secondly, when you're really working with hundreds of thousands of cells, sequencing to saturation becomes quite costly. So for the most part, this is not to replace classical single-cell sequencing, but a way to really enable applications with million-scale readout. We can also feed that into machine learning methods. In particular, we've developed essentially I wouldn't say an alternative, but kind of a different take on what is not graph neural networks, but takes the interpretability straight into the core hidden layers of the deep neural network, essentially doing deep learning on gene regulatory network models from the cell surface receptors all the way down to the transcription factors and their target genes to get to an interpretability. And then I already mentioned that we want to apply this in organoids as a readily perturbable system. And we kind of within the Human Cell Atlas, we try to provide a platform that makes such data readily available, essentially connecting the in vitro organoid, which is readily perturbable, with its in vivo complements. So that sums up my talk, and I would just have one slide of summary. I talked a lot about kind of epigenetic past and future of cells. So the epigenome allows us to kind of uncover this developmental record that cells keep among themselves. And this way we can reconstruct or developmental lineage trees. But epigenetic cell states also capture what a cell can do in the future, like I showed in the structure immunity setup. And what we're trying now is take this forward in a cell engineering perspective to make better cell-based therapies, to engineer CAR T cells based kind of on epigenetic programming to kind of better sustain the challenges of hostile tumor microenvironments, and to essentially become epigenetically programmed killer cells. With that, I would like to conclude and thank my team and all the collaborators who have contributed. Thank you.
And thank you very much for a wonderful talk. So we have time for questions if people want to start, please.
Thank you. Very nice talk. Just a technical question on the last bit, the sci-fi RNA-seq. If sequencing costs are the problem, have you started thinking about these new probe-based sequencing methods? Like, for example, the new 10x single-cell fixed profiling kit, right, uses, I think, about 19,000 probes to target mostly coding transcripts and remove things like mitochondrial or ribosomal transcripts. Can you still think of a way of having, say, 5,000 genes with a lot of information that allows you to get away with much less sequencing?
Yes, there is the computer scientist answer to that and the biologist answer to that. The computer scientist answer is, since everything is connected, the information is there, and you can take any 500 random genes and you will get as much information out of that random genes put in the right autoencoder, whatever is set up, as you would get from 500 selected genes. So it seems that, and people have shown that in the end, there are no kind of, if you are in the range of 500 or 1,000 genes, there are not really that many uninformative genes, like completely boring, unconnected, because it's such an interregulated system that somehow everything is captured somewhere. This being said, the biologist's answer is, of course, let's do that. Because if that's my 50 genes that everyone in the community knows are important in that particular system, it's much, much easier to convince myself, my colleagues, etc., that here's what's happening, and people have done this. So there is a version of CropSeq developed by the MBL that uses a kind of enrichment step to really go on a handful of target genes. And that can be a big cost saver, definitely.
Right, we have time for one more question.
Thanks, Christoph, for sharing your sort of also vision on the screening. And actually, I didn't know that review, so a very nice one. What's your take? What's missing on the computational side? So at the moment, right? Let's say you come up with a bunch of targets. Should we need something where we can now integrate this in a bigger picture? Should we just sort of deal with lists of differentially expressed genes, or do we maybe at some point need to go back to, let's say we build a transcription and regulatory network that then is sort of at the core for the next experiment? So how, let's say all of this works at some point, how do you want this to be sort of packaged into something that people would use downstream then?
I think more and more we should work on gene sets and kind of biological pathways. The simplest thing is you can just calculate average signaling, average kind of expression levels on kind of gene sets. But even better, we would somehow move kind of weighted averages through a gene regulatory network and try to pull out maybe not 20,000, but 1,000 key measurements of what's important that are ideally interpreted. And then we can do the perturbation analysis with these readouts. And if we were kind of, essentially what I'm saying is, let's define a language that is not a single gene language of how cells answer to a lot of things, because my guess is cells will answer in kind of reproducible ways or in kind of like non-unique ways. So there's cells will use a set of building blocks how to answer to stimuli, and we need to somehow get to those building blocks and essentially get to like a mesoscale understanding that is not differentially expressed genes, but it's also not cell type or cell state, but something in between that can easily through deep learning or whatever be translated up to the cell state and down to the single cell identity, but having this kind of endophenotype of what a cell's kind of core identity is at that point.
So that links a bit to Omar's targeted question in a sense. So would you also recommend to have epigenomics measured, let's say attack at the same time in a screen as well?
So we find epigenetics, we would actually rather do DNA methylation than ATAC-seq. ATAC-seq is just a compromise because DNA methylation is just hard to do at scale because bisulfite is such a toxic thing. So we cannot easily combine this. Soon we will be able to, but at the moment we cannot easily combine this with our single-cell RNA-seq readout at scale. Epigenetics, DNA methylation more so than ATAC-seq, but also ATAC-seq, gives us this past and future dimension, whereas the transcriptome really tells us what the cell is doing now. And if we can integrate these two, we have a bit of a time dimension. I think what people haven't really done is doing a meaningful integration between short time, which kind of you can somehow infer from single-cell RNA-seq data, and kind of long time, the developmental time. And bringing these two together, probably one would have to have some time series where one has kind of slow processes and fast processes with dense time series profiling. And that would give the training data to start being able to model kind of short-term processes and kind of that are in the order of hours to days, and longer-term processes that are in the processes of weeks that will probably follow a different way of, or different grammar of reacting to stimuli.
All right, let's thank our speaker. Thank you very much.